So chapter 26 is environmental microbiology. Okay. This is ecology. This is your interconnecting web of life, how the whole planet interacts with microbes all the way up to human beings. It's microbial ecology. It's the microbes in their natural habitat. Okay. It's applied microbiology, the study of practical use of microbes in food processing, industrial production, and biotechnology, how we actually use these microbes we've been studying about. Ecology studies and deals with biotic factors, which are any living or dead organism that occupy an organism's habitat, and abiotic factors, which are your non-living components. Okay. The ecosystem is the collective interaction between all the organisms and the surrounding physical and chemical factors in a given location, is an ecosystem. The biosphere is all physical locations on Earth that support life. That's our biosphere, the whole thing. It can be divided into a hydrosphere, a lithosphere, and an atmosphere. The hydrosphere is water. The lithosphere is soil or dirt. The atmosphere is your air. Maintains and creates conditions of temperature, light, gases, moisture, and minerals required for life processes, all things necessary to sustain life. So if you look at the Earth, this is the total Earth is your biosphere and the atmosphere surrounding it. The lithosphere is the soil. Like I said, the hydrosphere is the water, and the atmosphere is the air surrounding it. We got tropical forests. We have biomes, which is your temperate deciduous forest, the taiga, and the tundra. Okay. Organization of it is in biomes, which is particular climatic regions. So everything has specific climate. It's defined by large assemblies of organisms adapted to distinguished geography and climate characterized by dominant life forms, usually plants, communities, or association of organisms that live together, okay, and exhibit well-defined nutritional, behavioral interrelationships. And the definition of population is a group of organisms of the same kind. That's a population. So here's your ecosystem we talked about: lithosphere, hydrosphere, and atmosphere. We have the biomes, different climate-controlled areas. You go from south to north, you have different ecosystems as it goes from the soil and the water merge together. Different organisms in that, different populations inside that. And each population can be brought down to an individual organism. Okay. You have a habitat. Habitat is the physical location in the environment to which an organism has adapted. A niche. A niche is an overall role that a species or population serves in a community. It's a specific area that each one is defined where its habitat occurs. Its nutritional intake, position in the community, and the rate of population growth is all determined by its niche, the little area it's carved out for itself, basically. Okay, so a food chain, or basically an energy pyramid. It's not really a chain. It's not a single line like linked together. It's more of a web where it all interacts. But it's just called the food chain, and it's feeding levels and trace of flow and quantity of available energy from one level to another. Okay, you have producers, consumers, and decomposers. Producers provide energy, okay, that thrive the trophic pyramid. You have autotrophs, photoautotrophs, and chemoautotrophs. It depends how they make the energy and put it into the system. Consumers feed on the other living organism. Those obtain energy from the system and present the organic substrate they contain. Right? And then they got decomposers, which break down and absorb the organic matter of dead organisms, primarily your microbes. So you got producers to make the energy, consumers that feed on that energy, and decomposers that break down it once those organisms die, back and put back into the circle. Okay. So here's your energy and nutritional flow in ecosystem. You can see the autotrophs and the heterotrophs. Okay. Those are your producers. Then you got consumers and decomposers of that. You have an energy source. We had CO2 and water, makes up sugars and oxygen. Okay, so you have primary producers. From the primary producer, you got the ones that primarily feed on that. So they're primary consumers, secondary consumers, tertiary consumers, or quaternary consumers. So like plants produce the energy, like the cows feed on the plant, we feed on the cows, et cetera. We go up, each consumer goes up as it extends up the chain. And then once the consumers die off, they decompose, break it down, and feed it back into a CO2 source, carbon source. Okay. What we can talk about is the major roles of microorganisms. Primary producers. Primary producers are photosynthesis and chemosynthesis. Algae, bacteria, sulfur bacteria, chemolithotrophic bacteria in thermal vents. Those all produce energy. Then we start consumers, which are predation. Okay, free-living protozoa that feed on algae and bacteria. Some fungi that prey upon nematodes. We got the decomposers we talked about, which degrading dead organisms and waste. Soil saproprobes, primary bacteria and fungi, degrade cellulose, lignin, and other complex macromolecules. We got mineralization of the organic compounds, soil bacteria that reproduce organic compounds to inorganic compounds like CO2, so we can circle back and use it again. Okay. Cycling agents for biochemical cycles. We got recycling compounds containing carbon, nitrogen, phosphorus, and sulfur. Yeah, all the ones that our amino acids are made of that build our building blocks for proteins for life. Okay? And include hydrogen in that too. Hydrogen is just gas in the air. But it's basically carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. So you got specialized bacteria that transform elements and keep them cycling, okay, from the biotic to the abiotic and back to the biotic process. So from living to the dead and circle back, so incorporate back into energy and back into the living. And we have parasites, which living and feed on your host. We talked a lot about parasites, the viruses and bacteria we talked about. Viruses, bacteria, protozoa, fungi, and worms that play a role in population control. Okay, so the food chain or the food web. See, energy does not cycle. Energy cannot be created or destroyed. First law of thermodynamics. As energy is transferred to the next level, a large portion of the energy will be lost that can be not used out of the system. When energy is lost, it's usually given off as heat from the system. A food chain provides the most basic image of feeding pathway. It's simplistic, but it can help us understand how to identify the types of organisms that are present at a given trophic level in the natural setting. Okay, so here's the food chain. Diatoms are producers, produce energies. Protozoans feed on that. Secondary consumers feed on the protozoans. Insect larvae feed on the secondary consumers, and then the minnow feed on, as a quaternary consumer, feeds on the tertiary consumer. So it goes up and up and up, and then we feed on the fish. Okay, so it's basically, that's a simplistic food chain. A food web is a multi-level food chain. It represents more accurately the feeding relationship of a community. It doesn't go line by line by line like that. It represents the action, nutritional structure of a community and helps identify feeding patterns. So that's more like what it looks like. Okay, you can see how different things form other things. The sunlight feeds on the algae. Algae produces energy. Paramecium can go, bacteria can feed on the paramecium, or it can go up to the amoeba, or can come across. And it's more of a web that comes up with a food chain. But you can see this, the producers, the primary, the secondary, tertiary, and quaternary levels of the food web. Okay, so dynamic interrelationships based on nutrition and shared habitat is mutualism, which is association benefit to both members. So both members benefit on the relationship in the food chain. Commensalism, one member benefits while the other does not benefit, but it's not harmed either. It just has no effect on it. It's just used in the system. That centrifism is a metabolic process of one or useful nutrients for another. So in other words, once something uses that energy, its byproduct can be used by something else to feed it. So that's when the metabolic products of one are useful nutrients for the other. That's centrifism. Okay, then synergism is two usually independent organisms cooperate to break down a nutrient. Neither one could have metabolized alone. So you have to work together to break down a product to be able to use it. So the different interactions between organisms in a community. Okay, you have parasitism. Okay, one derives its nutrients or in habitat from a host that is usually harmed in the process. So just like a medical parasite that causes disease in humans or in other organisms. Okay, it uses the nutrients from one organism and it breaks it down, uses energy, and harms the other organism in the process. Competition. One member gives off an antagonistic substance that inhibits or kills susceptible species sharing its habitat. So one of the byproducts is toxic during the part of the species. It competes for the food source. A predator, consumer that actively seeks out and ingests live prey. And scavengers, feed on a whole different variety of food sources, not just one. Okay. So the natural recycling of bioelements is the process by which these bioelements and building blocks of protoplasm are recycled between biotic, which is your living, and abiotic, which is your non-living environments. Essential elements are cycled through the biological, geological, and chemical mechanisms. So it's a bio-geochemical cycle. Okay, includes biology, the living organisms, geology by the nutrients in the soil, nutrients in the air, the atmosphere, and the water, and then the chemicals that are present there. Microorganisms remove elements from their inorganic reservoirs, okay, and convey them into the food web. The Gaia theory, the biosphere contains a diversity of habitats and niches that are favorable to life because living things made it that way. We created that situation. We have trees that take in CO2 and water and produce sugar and oxygen. Okay, we take that in, we use that in our systems for metabolism, respiration, and we give off CO2 and water. We break down the sugar and by using oxygen metabolism through aerobic respiration, and then we give that back off to the trees as CO2 and water to be used again for conversion back into energy. So we created that niche or those habitats. We feed each other. Okay, so the quantity of available nutrients from the lower levels of the energy pyramid to the higher ones, does it increase, decrease, remain stable, or cycle? So the quantity available, what's available? We got a whole bunch of nutrients available, and then we use it to make energy. And as it goes up the chain, there's less and less because we use some of that up. So as you go up the chain, are you going to increase, decrease, or keep the same, or cycle the energy pyramid, the available nutrients? They have to decrease because we're using them as it goes up the chain. Okay, so it has to be decreased. Yep, and it is decreases. So the quantity of available nutrients decreases from the lower level of the energy pyramid to the higher ones. As you go up higher, the amount of available nutrients becomes less and less and less because we've used them up in maintaining the other species below it as it goes up the chain. Does that make sense?
Kind of. Yeah, it does. Because you're using the, like, as the little critters feed on the, like, on the
Yeah, you're right.
like we get less and less, I guess, nutrients because
Well, less are available. So we got to eat the other ones that have already consumed those. But there's less available nutrients.
okay.
Does that make sense?
Yeah.
All right, perfect. So we have atmospheric cycles. We have a carbon cycle, nitrogen cycle to recycle the carbon nitrogen back into the biosphere, back into living systems. So the carbon cycle, intimately associated with the energy transfers and trophic patterns of the biosphere. Carbon is recycled through the ecosystems by photosynthesis. Photosynthesis where plants take in CO2 and water to make sugar and oxygen. Respiration, fermentation of organic molecules. Like we make alcohol. Another thing, when ferment it, limestone decomposition breaks down to CO2 and methane production. Methane gas produces CH4, produces carbon. Principal users of atmospheric CO2 are photosynthetic autotrophs. Okay, those that make sugar and oxygen from CO2 and water. Carbon is returned to the atmosphere as CO2 by respiration when we breathe it out. We burn the sugar by metabolism using oxygen, producing CO2. Okay, fermentation produces carbon dioxide. That's when you make alcohol, you make wine, you make champagne, all those bubbles in champagne. All that is CO2 being released by fermentation. So all the fancy bubbles in champagne you're paying for is just wine with carbon dioxide being trapped in it. That's what causes pressure, and that's what causes the pressure in Coca-Cola. When you open a can, you get that pop. That's CO2 being released. Okay. Decomposition of marine deposits and burning fossil fuels all cause CO2 production. Methanogens reduce CO2 and give off methane gas, CH4. Okay, here's a basic summation of the carbon cycle. Volcanoes, respiration, combustion, respirations of organic carbons, other respiration of trees, all give off CO2 in the atmosphere. We take that CO2 back into photosynthesis with the plants and algae, and we produce back in, we feed it back into the system. Okay? Those respirations, they take in the oxygen and the sugar produced by the photosynthesis and reproduce CO2 back in the atmosphere. Okay? The nitrogen cycle. Nitrogen gas, most abundant gas, roughly 80%, 79% of the air volume is nitrogen. Our atmosphere is nitrogen. So most of our oxygen gas is nitrogen. Nitrogen cycle involves several types of microbes. There's 4 types of reactions that occur with these microbes for nitrogen. Okay, nitrogen fixation, ammonification, Ammonia is NH4. Nitrification, nitrate is NO2. And denitrification, break down to nitrogen. Okay. Nitrogen fixation, atmospheric nitrogen is converted to NH4 salts, ammonium salts. Nitrogen fixing bacteria live free or in symbiotic relationships with plants. That's how we get nitrogen to go into NH4 so you can use it. Ammonification, bacteria decompose nitrogen containing organic compounds to ammonia. Ammonia is NH3. Okay. Nitrification converts NH4 plus, which is ammonium, to NO2 minus, which is nitrite, and NO3 minus, which is nitrate. Denitrification, microbial conversion of nitrate back to atmospheric N2 and completes the cycle. You got to produce what you use up or you're going to run out of it. Okay, so there's the nitrogen cycle. You see it's in the plants, the roots, nitrogen fixation. Okay, so atmospheric nitrogen goes into the plant, into the roots. Nitrogen goes to NH4. NH4 goes to NO2, which goes to NO3 minus, which goes back into the plant for respiration, which produces nitrogen back into the atmosphere. Okay. Nitrogen fixation in the root nodules. What happens is the symbiotic relation between rhizobia and legumes accomplish nitrogen fixation, which is a natural fertilizer factories. They basically take the nitrogen from the NO3, goes from the ammonium to the NO2 minus to the NO3 minus, and that gets fixed back into nitrogen gas and leaks back into the atmosphere. That fixes nitrogen. Okay. These are the rhizomes and the legumes underneath the plants that cause the symbiotic relationship that the bacteria adhere to the plant roots, and they allow for this nitrogen fixation to occur. Okay. There's the green part of the plant, which is your chlorophyll, which absorbs the sunlight, so photosynthesis can occur. And then you have little chloroplasts you can see on the screen there on light microscope. And you get sedimentary cycles, sulfur cycle, phosphorus cycle. We got to refine sulfur and phosphorus, which are also part of our biological system. Sulfur is part of amino acids, phosphorus part of amino acids. Phosphorus is part of our ATP, energy production in the bodies. Okay, so the sulfur cycle. Sulfur originates from rocks, oceans, and lakes. Sulfur exists in the elemental form as hydrogen sulfide gas, sulfate, and thiosulfate. Plants and microbes can assimilate only sulfite ion, SO4. And now it's required an organic source. Okay, amino acids, cysteine, cysteine, methionine. Okay, those are all different types of amino acids which we build our bodies from. There's 20 different building blocks of amino acids that we use to build all proteins in our body. Bacteria convert environmental sulfurous compounds into useful substrates, into useful sulfates. Okay, the phosphorus cycle. Okay, chief organic phosphate is PO4. It's from phosphate rock. PO4 is converted into a usable form, which is our phosphate ion, which we use to phosphate ATP, produce all the energy for the body. Okay, this is usable by the action of an acid. Sulfuric acid, H2SO4, is naturally released by some bacteria. The organic phosphate is returned to soluble phosphate by decomposers. They break it down. Here's your phosphorus cycle. Again, you start at the very bottom. You have consumers, organic phosphate, and producers organic phosphates. See that? Mineralization, you have mining and phosphatizing material produce the PO4 minus, which is a phosphate ion. Okay, and it forms into phosphate rocks, calcium, phosphate, and fluorine. Okay. And that's how the cycle feeds into the consumers at the bottom of the organic phosphate. Other types of cycling: introduction of toxic substances escalates cycling of elements. Some are converted to less harmful ones. Some persist and flow into the biosphere. Bioaccumulation is when a pollutant is accumulated by living things through natural trophic flow of the ecosystem, like mercury compounds. It's a natural flow of mercury coming, and it's a toxic to our bodies. Okay. Composition of the lithosphere. Remember, the lithosphere is the soil. Okay, soil is a dynamic ecosystem, part of the lithosphere that supports interactions between geological, chemical, and biological factors. Okay, it teems with microbes, serves as a dynamic role in biogeochemical cycles, which you talked about, as an important repository for organic detritus and dead terrestrial organisms. That's where the soil accumulates. Okay, you can see the soil here is a plant root. You can see the sand and the dirt around it, the water molecules around it, clay, silt, and all that. And you see the mycorrhiza hyphae, which goes around the root. Okay, you get fungi that goes around it. Mycorrhiza spores are what are fungi that allow nitrogen fixation to be associated with the roots of a plant. Okay. So in the lithosphere, the soil is, you have different areas. You have the humus, which is a rich, moist layer of soil containing plants and animal debris that's being decomposed by microbes. Okay. The rhizosphere, the zone of soil around the plant's roots, containing associated bacteria, fungi, and protozoa. And mycorrhizae, which is a symbiotic relationship formed between fungi and certain plant roots. The fungi fix the nitrogen around the plant's root, they absorb that, and they use it in the process of making amino acids for the plants. So it's a symbiotic relationship. And this is mycorrhizae that are associated with the roots of the plants as it grows around them. Okay, so root nodules contain what, which can do what? So in the roots, which system is present in the roots? Azobacter, Nitrosomonas, or Rhizobia, or Acillus?
Is
Which one of those
it
are
is it rhizib
Rhizobia? C,
oh,
right? Yeah, Rhizobia. That's the fungi that goes down the roots of the plants, and they fix the nitrogen back in the soil, producing nitrogen gas back into the atmosphere. Yep, and they're in a symbiotic relationship with the roots of the plant. So it would be C. Aquatic microbiology. So water is the dominant compound on the earth, occupies 3/4 of the earth's surface. 75% of the earth is water. Water is continuously cycled between the hydrosphere, the atmosphere, and the lithosphere. It's called the hydrologic cycle. Water goes from the soil into from the water to the soil to the atmosphere, and then back to the water, back to the oceans again. 97% of all the water on Earth is in the oceans. 2% are glaciers, groundwater, lakes, soil, rivers, all that. It totals 100%. So almost all of our water is in our oceans. Okay, hydrologic cycle, water evaporates, accumulates in the atmosphere, and then returns to the earth through condensation, precipitation. You have a cycle. Surface water collects in subterranean pockets, forms groundwater sources called an aquifer, and resurfaces through springs, geysers, hot vents. It's tapped as a primary source for quarter water for human consumption. Okay, and there's your water cycle. Okay, condensation goes down the atmosphere, then rains back down through precipitation, ice, snow, etc. You can see all the different areas where the water recycles through the water cycle there, the hydrologic cycle. Okay. Surface waters differ considerably in size, geographic location, and physical and chemical character. Okay, sunlight, temperature, aeration, dissolved nutrients content are factors that contribute to the development of zones. The lake is stratified both vertically and horizontally into several zones or strata. Freshwater zones, those are your vertical zones. Those are photic zones, profundal zones, and benthic zones. The photic zone is surface to lowest limit of sunlight penetration. So it goes from the surface to how far down the sunlight can penetrate. That's a photic zone. The profundal zone is the edge of the photic zone until the bottom, to the lake sediment, to the base. The benthic zone, organic debris and mud forming the basin, is your benthic zone. So those are all vertical zones going straight down. Horizontal zones go across now. Those are the littoral zone, which is your shoreline, which is shallow water, and the limnic zone, which is your open or deep water. So that's how it's divided up. Okay, you can see here there's a different life forms that inhabit the different zones. You can see the horizontal zones, okay, which is your littoral zone and your limnic zones, your deep water and your shallow water. Then your vertical zones, your photic zone, your profundal zone, and your benthic zone, where light penetrates it. Okay, so marine environments resemble profiles, okay, of lake, but has variations in salinity, different types of salt concentrations, the depth, temperature, hydrostatic pressure, and the mixing of it. Okay, it contains a zone called the estuary zone. Estuary zones where rivers, which was your freshwater, we talked about the freshwater zone, meets the sea. So those are zones that have a fluctuation or an inconsistency in the salt content. Okay, It's very high in nutrients in those areas. We find a lot of growth in those areas. Okay, tidal wave action suggests the coastal habitat to alternate periods of submersion and exposure. The abyssal zone extends to a depth of 10,000 meters, support communities with extreme adaptations, including halophilic, psychrophilic, barophilic, and anaerobic. That's the abyssal zone. Okay, the microbial distribution associated with sunlight, temperature, oxygen levels, and available nutrients. That controls what lives in different zones. Like I said, the photic zone, which has sunlight. Remember, that's from the surface of the water to the area where the sunlight stops penetrating. That's your photic zone. It's the most productive zone that contains plankton. Okay, phytoplankton, variety of photosynthetic algae and cyanobacteria that produce oxygen and do through photosynthesis. Zooplankton, microscopic consumers, filter fed, prey or scavenge, are used zooplankton. The benthic zone, remember the benthic zone was the soil underneath the water. Supports a variety of organisms, including aerobic and anaerobic bacterial decomposers. That's when things die in different zones, they float down to the benthic zone areas. Okay, this is your aquatic community. You can see the different cycles here, viruses, bacteria, we all talked about. These are dissolved organic materials, they come up, and you can see how they cycle into different zones. You have primary consumers, secondary consumers, tertiary consumers. Okay, so the primary producers are in the phytoplankton, the cyanobacteria. Lake profiles, lake bodies of standing water develop thermal stratifications. Stratification is just zones that are developed in the lakes. There's an epimelian, the hypolimnion, and the thermocycline. Okay, the epimelian zone is the upper region, which is your warmest region of the lake. The hypolimnion is the deeper, cooler water. The thermocline is that buffer zone between the warmest and the coolest. Okay, so between the epilimnion and the hypolimnion is a thermocline. That's what separates the two layers. Okay, and it's what this is the different layers of a lake profile. It depends on, and a lot of it depends upon the thermal conditions, the heat, the winds, things like that. But the epilimnion is the top layer, which is the warmest, then the thermocline, which isolates the two layers, and then the hypolimnion, which is the coldest of the layers. And different bacteria, different viruses, different organisms live in those different zones. Okay, the conditions live under different conditions that those zones exist at.
Like the warmest part of the lakes, I was reading that, like, what is it called? It's called like Naegleria valori.
Yeah.
They like to go in like the warm lakes.
Yep. So then they live in the top surface of it. And different fish live in the different layers. Like if you're looking at like the species of trout live in the hypolimnion layer. They live in extremely cold layers of the lake. Okay. Different fish live in the minnows live in the bluegill, live in the top layers in the warm waters of the lake. Okay. And the organisms are set for and adapt to the different temperature regions. So it's kind of interesting how those do this. And when you have like a big wind or a storm, those all get mixed up. All the nutrients get mixed up, and the layers can change up in there because you bring the nutrients up from the bottom, and the temperature zones get distributed differently when the winds come up until it settles back down. So currents brought on by temperature change cause upwelling of nutrients, rich bathenic sediment, and outbreaks of abundant microbial growth. So all of a sudden, you bring these nutrients up from the bottom. Now the layer, like the plankton that live on top, can feed like crazy and overgrow a lake. That's something you overgrow, like algae and stuff like that, like red tides. Red tides is really pretty at night if you go down the beach in Southern California, because when they break, you see this fluorescent glow over them. Okay? But it's the bacteria that have been swelled up by the different currents, and now they can feed and multiply. You can see the red discoloration of the tides during the day, as the plankton, the phytoplankton on the top, have more nutrients to feed on, and they get an overgrowth. And sometimes the overgrowth is very obvious. We'll see a couple more pictures of that coming on too. So the aquatic nutrients, the oligotrophic are nutrient deficient ecosystems. There's few microorganisms, but there's many bacteriophages that live in those zones. The eutrophication are addition of excess nutrients, naturally or by effluent from sewage, agriculture industry dump in different nutrients into those layers. Heavy surface growth of algae can occur, which is called a bloom, cuts off the oxygen supply, disturbs ecological balance. Only anaerobic or other factors of anaerobe survive. And you can see where the plankton and the algae can overgrow it because those minerals have been stirred up either by dumping stuff into the lakes, giving them extra nutrients somehow, and they continue to grow. They're not limited in, and they can take over that system on the top and surface of the water. They can cut out oxygen and other things. Okay. Potable drinking water. Potable means it's drinkable. Okay. So potable water is free of pathogens, free of toxins, free of turbidity, odor, color, and taste. That's water that we drink. That's our ideal goal when we're purifying water for our water supply, to make it potable. Most prominent waterborne pathogens are Giardia. We talk about Giardia causing hepatitis A, Cryptosporidium, which are protozoans, Campylobacter, Salmonella, Shingilla, Vibrio, Mycobacterium. These are different bacteria, hepatitis A, and noroviruses are all present. We have to get rid of those in our potable water. Most assays of water purity focus on detecting fecal contamination, which is indicated by E. coli bacteria or Enterobacter or Citrobacter. Okay. General indicators of water quality are standard plate count, which we count the number of colonies that develop, provide estimates of the total population. So we take a sample, put it in the lab, put it in aggregate plates, and see how many colonies develop. And from that, we can estimate from the sample size how many colonies are in the water supply. Biological oxygen demand, BOD, water with high levels of organic matter and bacteria have low oxygen content. Why is that? Because the bacteria and the other things have absorbed the oxygen because they're using them for metabolism. So the oxygen content decreases when the amount of bacteria and other organic matter in there increases. That's aerobic respiration. We use the oxygen for respiration, metabolism. We use a coliform assays, which are membrane filter methods. So after filtration, we take what comes out, is placed on a selective differential media. We incubate that, and the colonies are identified and counted as what's there and how much is there. Okay. The most probable number is MPN, which is presumptive, confirmatory, and completed tests. Okay. No acceptable level of fecal coliforms, enterocoli, viruses, or pathogenic protozoas in drinking water. So certain things we're allowed levels of. We cannot have levels of fecal coliform. Got a noise behind me. Oh, it's my daughter has a cat and she just crawled under the table. I'm not used to cats. So there's certain levels we can have and others we can have nothing of that. No fecal contamination, enterovirus, other viruses, or pathogenic protozoans in the drinking water. Okay, we have to have that zero tolerance for that. These are the different tests we can use. This is water analysis. You have the red one, the red, which is the normal, and then the white, which is the amber color on the right-hand side. And what it is the phenol red broth, something with lactose, they put sugar in it, is used as a preliminary indicator for the presence of coliforms, which you can't have any. The medium contains lactose and a pH indicator, tells what the acid is. Because as the bacteria feed on the lactose, it's going to change the acid content of the colony, of the media there. Okay, along with nutrients to support bacterial growth. So the presence of coliforms leads to the fermentation of the lactose, lowers the pH, gets it more acidic, okay, and causes the medium to turn from red, which is a negative test, to yellow, which is a positive test, which means bacteria are present. A small inverted tube called a Durham tube is placed within a larger culture to trap any gas that is produced. So we go back, I'm going to go back and look. If you go back and look, you see there's a little funny, there's a small test tube inside the bigger tube. That's going to trap any gas. Okay, and there you can see it. Those are the test tubes taken out, the Durham tubes. You can see on the left-hand side, the bubbles. Bubbles indicate that a gas has been formed. Okay, so the medium within the MUG is used to detect Escherichia coli, E. coli, which is fecal bacteria, bacteria from fecal matter. Selective agents inhibit gram-positive bacteria, while the MUG, which is the growth media, acts as a substrate that only E. coli can hydrolyze, produces a fluorescent product. As seen on the left side, we saw that it was a fluorescent glow. We saw how it kind of fluoresces on the left-hand side there. Probably seen in the test tube on the left. That's a negative reaction is seen on the test tube on the right. So we have a positive reaction on the left, which you have fluorescence and starts to bubble. On the right, we have not a lot of bubbles and no change in the media. So typically, a positive reaction. The gas from fermentation is also observed. So not only does it fluorescence, but you'll see the gas bubbles being formed. Okay. Another way is through the coliform, to determine coliform contamination is a water analysis through a coliform water analysis of it through a container. This is through filtration. Okay, so the membrane filter technique, the water sample is filtered through a sterile membrane, the filter assembly and collected in a flask. The flask is then removed, placed in a small petri dish containing different selective media. Okay, and then we incubate it on the MFD endomedium, colonies of the E. coli, often yielding noticeable metallic sheen. Okay, the medium permits easy differentiation in various genera of coliforms. And then you have a grid pattern where you set it up, can be used as a guide for rapidly counting the colonies. Okay, so the floating assemblage of microbes and animals that drifts on or near the surface of large bodies of water is the what community? Which one of those, the abyssal, the benthic, the littoral, or the plankton, which one of those exists on the surface? The floating assemblage of microbes. So what floats up, floats on the surface. Okay, or near the surface of large body of water. Which one is it? Which of those four?
Was it the plankton?
It was the plankton. That's one that exists and can overgrow with excessive nutrients.